Welcome to the Chemistry, Biology and Math Revision Hub. Today we're doing the A-Level Biology topics. This is cellular respiration. Beginning with topic 7A1, respiration in cells. This is a process by which energy from food molecules is converted into ATP. This can be aerobic or anaerobic. Aerobic respiration takes place in the presence of oxygen and it occurs in the mitochondria. During this reaction, the respiratory substrate is going to be converted into carbon dioxide and water, as well as the release of large amounts of energy. The volume of oxygen used and CO2 produced can depend on the level of activity of the organism, as well as other factors like temperature during the respiration process. This is the layout of the whole process of aerobic respiration. This occurs into two distinct phases. One is going to be glycolysis, which occurs in the cytoplasm, and this is going to be enzyme controlled. The respiratory substrate is going to be broken down into pyruvate. And during glycolysis, small amounts of ATP are going to be produced. Then pyruvate is going to enter into the link reactions. These occur in the mitochondria, and then into the Krebs cycle, the electron transport chain, and so on. So the final stage, which is oxidative phosphorylation, requires oxygen in order to produce ATP. In 7A2, we're going to talk about glycolysis and anaerobic respiration. During glycolysis, we can break it down into 4 steps. The first step is phosphorylation. In this case, 2 ATP molecules are going to be used to phosphorylate either end of the glucose molecule. We can see this phosphate and that phosphate, and 2 ADPs are going to be produced in the process. Then the next step is going to be lysis, in which the 6 carbon molecule is going to be broken down. We produce a 3 carbon that is phosphorylated on one side and a 3 carbon phosphorylated on the other side. Then some inorganic phosphate is going to be delivered. This is not from ATP, but it's going to be delivered, and the end that was not phosphorylated is going to be phosphorylated. Phosphorylation makes organic molecules to be activated in order for them to participate in certain reactions. After phosphorylation, we go to oxidation, where an electron carrier molecule is going to be reduced, as this is going to be oxidized. But also 2 ADPs are going to be converted into 2 ATPs in the process of ATP formation. So each 3 carbon molecule is going to produce 2 ATPs. In total, we have produced 4 ATP molecules from the 6 carbon molecule. However, 2 ATPs were used up in the process, so the net produced ATP is going to be 2. ATP production in glycolysis is produced through substrate level phosphorylation, and it does not require oxygen. During cellular respiration, most of the ATP is going to be made through the final stage, which is oxidative phosphorylation, with the help of the electron transport chain. Here, electron acceptor molecules like NAD as well as FAD, which are coenzymes, are going to accept hydrogens to be reduced, and the reduced products are going to carry the electrons as well as the protons towards the electron transport chain. Anaerobic respiration occurs during insufficient supply of oxygen. In humans or in animals, pyruvate produced is going to be converted into lactic acid, or sometimes called lactate. And then in plants, it's going to be converted into ethanol as well as carbon dioxide. And in both processes, ATP is going to be generated, but this is small amounts of ATP. Like we can see here in animals, the glucose is going to enter the cell. We see it in the cytoplasm, and here it's going to be converted into pyruvate, like we saw previously through the process of glycolysis. ATP is going to be used and ATP is going to be generated, and we saw that per 1 glucose molecule, 2 net ATPs are going to be generated. And this produced pyruvate cannot enter the mitochondria. It's going to be converted into lactate. In the conversion of pyruvate to lactate, the reduced NAD is going to be used and it's going to be converted into the oxidized version of NAD. So pyruvate becomes lactate. However, this produced lactate is going to accumulate in the body, and most of the times it leads to muscle fatigue as well as soreness. And in plants, the pyruvate is going to be converted into ethanol in the process of fermentation. Here, still small amounts of ATP are going to be produced. Carbon dioxide is also going to be produced. So we can see the equations for both processes. In animals, we can see glucose is converted to lactic acid, which is lactate. Some ATP is produced. And in plants, we can see glucose is going to be converted to ethanol and carbon dioxide. This occurs in plants as well as in yeast cells. In topic 7A3, the Krebs cycle, the products from glycolysis will enter the link reactions before they go into the Krebs cycle. Here, the 3 carbon compound pyruvate is going to cross through the mitochondrial membrane from the cytoplasm into the mitochondria. This process is called decarboxylation, resulting in the formation of carbon dioxide molecule. At the same time, the pyruvate is going to be oxidized, producing reduced NAD. So we can see the reduced NAD formed here in the process of dehydrogenation, as well as carbon dioxide produced and the 2-carbon compound. Then this 2-carbon compound is going to join with coenzyme A to form acetyl-CoA. So the 3-carbon molecule that entered is now a 2-carbon molecule that enters into the Krebs cycle. And in the Krebs cycle, this is a series of biochemical steps that lead to the complete oxidation of glucose. And in the end, we're going to produce carbon dioxide, as well as some electron carrier molecules like reduced NAD and reduced FAD, which will go into the electron transport chain. During the Krebs cycle, each individual step is going to be controlled or catalyzed by specific enzymes. And these reactions occur in the mitochondrial matrix. ATP is going to be produced against substrate level, and carbon dioxide is going to be produced. So for example, here we have the 6-carbon compound, which is a result of the 4-carbon reacting with the 2-carbon. So when the 6 is formed, here it loses a carbon dioxide molecule and it becomes a 5-carbon compound. This process also involves formation of reduced NAD. Again, we see the 5-carbon is going to be converted into a 4-carbon by the loss of 1 carbon dioxide molecule. But here we produce 2 reduced NADs and 1 reduced FAD, and all these are going to be carried to the electron transport chain. In topic 7A4, here we are looking at the electron transport chain. This is where oxidative phosphorylation takes place with the help of chemiosmosis. We can see the components of the electron transport chain, component 1, 2, 3, 4, or you can call them complexes, are positioned within the inner mitochondrial membrane. And we can also see there is a molecule here called ATP synthase. The reduced electron carriers are going to deliver the electrons to the first complex. We can see reduced NAD delivers to complex 1 and reduced FAD delivers to complex 2. So when reduced NAD delivers to complex 1, the proton is going to be lost into the matrix, but the electron is going to be delivered to the first complex. And since these complexes within the electron transport chain are arranged at different energy levels, the first one being at a high energy level, so the electron is going to transition from that to the next, losing some energy, and as it transitions through all the complexes, energy is going to be lost, and the lost energy is going to be used to actively pump the protons from the mitochondrial matrix into the intermembrane space. These protons are going to accumulate into that intermembrane space, and they have no pathway to come back. So that accumulation creates an electrochemical gradient, a proton gradient, as well as a pH gradient. So the protons have to return through ATP synthase. Because they are higher in energy while in the intermembrane space, as they return through the ATP synthase, energy that they possess is going to be used to combine ADP and the inorganic phosphate in order to produce ATP. So ATP molecules are produced in that manner. And as the protons return inside, they're going to have less energy, so they can be combined with the electrons coming off from the electron transport chain, as well as oxygen, in order to produce water as the final product. Lastly, topic 7A5, respiratory substrates and respiratory quotient. Here we're going to be looking at the components that can be respired in the cell and the significance of which components that are respired. So starch can be converted into glucose. We know that is digestion breaking it down. And this glucose can be phosphorylated in order to produce the 3-carbon pyruvate. This pyruvate is going to enter into the mitochondria, where it's going to lose a carbon or carbon dioxide and then react with coenzyme A to produce acetyl-CoA, which enters the Krebs cycle. However, also glycogen in the body can be converted into glucose, and then it can go through the phosphorylation process. Also, the metabolism of proteins involves proteins being converted to amino acids. These amino acids can be converted into pyruvate or acetyl-CoA. Also, the other side, glycerol, can be phosphorylated, or it can be converted into fat. And this fat or the fatty acids are going to be converted into acetyl-CoA, which can be respired. So basically, this is a flowchart of how different molecules can be respired or can be used as respiratory substrates in the process of aerobic respiration. When we talk about respiration, we also talk about the respiratory quotient. To calculate the respiratory quotient, we need to measure the volume of carbon dioxide produced and the volume of oxygen used by an organism within a specific period of time. And then we can calculate respiratory quotient, which is equal to the volume of carbon dioxide produced divided by the volume of oxygen used. When we look at the value obtained from the RQ calculations, we can be able to predict which kind of foods have been oxidized in the body of that organism at a particular time. For example, carbohydrates can give an RQ of 1, fats can give an RQ of 0.7, and proteins about 0.9. If the RQ of an organism is greater than 1, then anaerobic respiration may be occurring, because that means there is more carbon dioxide produced compared to the oxygen used up. So that tells us there is more anaerobic respiration going on in comparison to aerobic respiration. So this brings us to the end of topic 7a, which is cellular respiration. Thank you for being with us. Do not forget to subscribe. See you in the next video. Bye-bye.